the overview. We're going to cover a lot about nitrogen assimilation. We don't do any of this, and also we don't do any sulfur assimilation, but in the biosphere, how all these things are made is extremely important for life on this planet. So we're going to cover all that. And then we're going to see how the amino acids specifically are made and how they're broken down. Let's see now. All right. So in terms of our diet, okay, we've discussed how we get our carbohydrates, mostly glucose, what happens to it. We finished in the up to exam 3, you know, what happens to fatty acids, how we break them down with beta oxidation, the mitochondria, and so on and so forth. But we do eat a lot of protein, and we do break down even our own protein, our own muscle protein, when we sleep, for example. And these amino acids are used as food, and they enter at various points in these fundamental central metabolic pathways that we discussed, mostly the TCA cycle. So they provide, it's an excellent source actually of providing all these intermediates that we need. So that also happens. But before we get to all this breakdown and synthesis of amino acids, we have to talk about something more basic, which is the nitrogen cycle on a planetary scale, right? So when we break down compounds that contain nitrogen, and those really are amino acids and nucleic acids, eventually, I mean, we excrete in various forms, but eventually it's going to be broken down to ammonia. Not necessarily by us, but by microorganisms that pick up our urine and decompose it even further. So what happens to this ammonia? This is nitrogen that's highly reduced, okay? This highly reduced nitrogen will be picked up by microorganisms. This is mostly a prokaryotic, not mostly exclusively a prokaryotic universe, all of these reactions, except this black thing that we and animals do. So we and other animals just contribute ammonia to the whole thing, but we have no input in how everything gets recycled back. So this highly reduced form of nitrogen, which is ammonia, will be oxidized. So these electrons will come off ammonia, and you can get an idea of what will happen to them. Okay, so what's good for these organisms is that they have a source now that can do this, to oxidize ammonia. This nitrification, the nitrifying organisms, they can use ammonia as an electron source, okay, and power their electron transport chain. And when that happens, they can power their batteries, of course, and they can make ATP. So that's their motivation. That's what they get out of it. In the process, of course, they recycle ammonia back to the biosphere here. So this happens in steps. Well, it can happen. It chemically definitely happens in steps. It was thought until very recently, I'll update you on this recent development, that specific organisms do each step of these ammonia oxidation reactions, which really are two major steps. First, ammonia gets oxidized to nitrite, and then nitrite gets further oxidized to nitrate. And then this is the nitrification process. And this always happens in the presence of oxygen. So this is an oxic environment. So you need oxygen for all these reactions, okay? Which is kind of obvious, because here in place of protons on nitrogen, we have the input now of oxygen. Now the reverse reactions, however, happen in the absence of oxygen, and they can only happen in the absence of oxygen in an anoxic environment. So the denitrifying organisms, other bacteria, take up nitrate and convert to nitrite, and then we have some intermediate ones, and all the way bring it back down to molecular nitrogen, dinitrogen, which is, of course, now the most abundant gas on this planet, right? I mean, 70% or so of our atmosphere is made of dinitrogen in this form, which is extremely inert, chemically speaking. It doesn't want to react with anything. Another interesting, that's also a very recent development, source of this dinitrogen is not only these purple arrows, but it was figured out recently that certain organisms, especially in the oceans, they can combine nitrite with ammonia to give dinitrogen. Now, this is chemically inert, and it cannot enter the living organisms again. I mean, they cannot really use it until somebody, so we cannot use this dinitrogen to make amino acids or nucleic acids, okay, DNA, RNA, and so on and so forth. So somebody has to fix it, has to put it into the cell ready for these chemical reactions, and that's always ammonium ion, okay, ammonia plus an extra proton, it's positively charged. This process of reintroducing now the inert dinitrogen from the atmosphere all the way into a form that can be assimilated by living organisms, such as plants and so on and so forth, is called nitrogen fixation. And it's an extremely important process. Okay, and without it, again, there won't be life. So we'll see how that happens too. But let's start with this nitrification process and the first step, how we go from ammonia to nitrite. So nitrification, again, is the biological oxidation of ammonia into nitrites first and then nitrates. This is a prokaryotic business. There are specific nitrifying prokaryotes that will do this. The first reaction is AMO, ammonium monooxygenase. So it will take ammonia, oxygen, and electrons. So, and then here we have the input of electrons, okay, but you'll see how the extra electrons will come off in the next reaction. And the first product you get is hydroxylamine. So it's just an amino group with a hydroxyl group. Then hydroxylamine will react in a reaction catalyzed by hydroxylamine oxyreductase. How? So we have the AMO and HAO enzymes here. Then you get nitrite, okay? And you can see here protons and electrons. And that's a hint that this can participate now in an electron transport chain. The species that can do this, there are this genera of bacteria, but also recently some archaea has been found to do this, to be able to do this to fundamental chemical reactions. The way it looks, the AMO reaction, the ammonia monooxygenase, takes 2 electrons from Q, okay, and that is necessary in order to plus oxygen, of course, and ammonia. Now, with these electrons, you can generate hydroxylamine. And then the HAO reaction will take place, and hydroxylamine will be converted to nitrite. And very importantly, the 4 electrons net that we have here can enter an electron transport chain with a cytochrome C, Q enzymes, and these all important electron transport complexes that can, in the process of moving the electrons, okay, can also pump protons. Okay, and that's the point for these organisms. They can actually use this source of electrons, ammonia, and power their batteries, in a sense, right? So it's food for them. In the second step, okay, so we saw how we go from ammonia to nitrite, from nitrite to nitrate, okay. The nitrite will be oxidized further. This species can do this. The reaction is catalyzed by nitrite oxidoreductase. And again, it's the same principle. We have the oxidation of nitrite, we get the nitrate, and in the process, okay, you get some extra electrons and protons getting pumped, so you can power your ATP synthase. So it's because of these features, these don't look, I mean, in principle, they look like our electron transport chain. The enzymes are different, but the chemical principles are the same, the way they are set up. And it is this realization by Peter Mitchell with the chemiosmotic hypothesis, how all these seemingly diverse phenomena, okay, got unified, okay, with the electron transport chain and the chemiosmotic hypothesis. So there are many ways organism life will explore every way possible to pass electrons along a membrane and at the same time pump protons out so it can power this most ancient ATP synthesizing machine, and ATP can be made. So as long as you can plug an ATP synthase onto a membrane and move electrons across and couple that with the transport of protons, you're done. That's the whole point of the chemiosmotic hypothesis. This is a cycle here. It's in reverse. Okay, we start from ammonia, and it's so in a clockwise fashion. The way we had it, it goes counterclockwise, and it was thought that different bacteria go from ammonia to nitrite, and a different set of bacteria goes from nitrite to nitrate. But I think last year it was discovered that there are some bacteria that can do both. So both steps can happen in one organism, okay, as opposed to separate organisms catalyzing each one of these two steps of nitrate formation. So this bacterium is called Comamox bacterium. It has been coined. So we are here now, okay, so we nitrified ammonia, okay, ammonia is now fully oxidized in the form of nitrate. How do we get now this dinitrogen? That's the point, and that has to be reduced, okay? So electrons have to be found to reduce nitrate to dinitrogen. By the way, too much nitrate is not good. It's toxic. It has to be denitrified. The bacteria that do what we're about to discuss have a very, very important role in maintaining a healthy planet. Again, they have been even introduced in some waste sites. It's a major concern in areas with high pollution to denitrify them. Here is the process, almost the reverse. Okay, from nitrate you go to nitrite, and then you go to these forms, nitrous oxide, for example, before you can get to dinitrogen. So electrons will be needed, and a reductase will be needed. And as the name implies, you start with nitrates, you need a nitrate reductase, you give it the electrons. And it's a molybdenum-based catalysis. That's kind of unusual. And once you make the nitrates, then you need to make the nitrites. Even more electrons are needed for this. And it is this form of ammonia of nitrogen that can stay in the cell and get assimilated. So you can go all the way to this step. But notice what you need. You need an enormous amount of reducing power. So you can spend a lot of NADPH if you need to do this. Or if you have the ability to capture the energy from the sun, you can use a photosynthetic mechanism to provide this massive reducing power that will allow you to convert nitrite to an assimilated form of nitrogen, such as ammonium. So remember, this is a slide from the photosystems. It's photosystem 1 is a very powerful reductant. It transfers electrons to ferredoxin. Okay, that's what it does. And you can use the reduced ferredoxin in any way you want. You don't have to make NADPH. You can reduce other things. So you don't have to necessarily to reduce NADP to NADPH. You can reduce, in this case, nitrite, right here, and make ammonium. So that's one source of reductant, and the difference is right there. That's a reduced ferredoxin. Instead of adding to it NADP, giving it NADP to reduce NADPH, you can give it nitrite, and make ammonium. And that illustrates a bigger picture here, which is, it's just one example of the uses of ferredoxin. You can use it for nitrogen assimilation. You can use it again in photosynthetic reactions that we have seen previously, or all kinds of reductions that are so important for life, whether it's sulfur assimilation too, for example, that we will see in a minute. All right. So that's one way. You can go from here all the way to ammonia. But a key issue here is to how is the dinitrogen, again, that we see in the atmosphere, how is it generated? So it turns out that the Anamox reaction, which was discovered, I think, in the 80s, generates at least half of the nitrogen in our atmosphere. So in this case, what you get, you combine is ammonia and nitrite, and you get dinitrogen, okay? So the two atoms of nitrogen come together and to make the molecular nitrogen that we see here. And as I said, that generates about half of all nitrogen in the oceans and the atmosphere. So how does that happen? We've seen some of these reactions, okay? In this case, so ammonia ion from inside the cell, from outside the cell, sorry, comes in through the periplasm, this bacteria, and then it comes in through the cytoplasm, becomes ammonia, and then gives rise to hydroxylamine. So we have seen this part of the reaction, how ammonia can give rise to hydroxylamine. It was in the nitrification, the very first nitrification reaction. This hydroxylamine now will combine with nitrite, and that's a new reaction, and will generate hydrazine. And of course, there is an electron transfer happening here, so that's what makes it possible, okay? And eventually, the electrons from hydrazine will be transferred on electron transport chain. The bacterium does something useful with them, as we know, and the product will be nitrogen, molecular nitrogen. So the way these Anamox bacteria are doing it is to reverse this Howe reaction we discussed. Here we had seen how from hydroxylamine you get to nitrite. That was the very first step in nitrification. But these bacteria do it in reverse, okay? And that's what they have to do. All right, so we described Anamox. We see how we get a lot of dinitrogen. And the last part we need to do is to cover this red arrow. We got there all the dinitrogen, but how do we actually fix nitrogen in a form that can be taken by cells such as plants, for example? And once you put it into plants, then you can feed everything else, all the animals and so on and so forth. So super important reaction, and that converts the inert dinitrogen into ammonium, and this, or ammonium more accurately. Only bacteria can do it. Okay, this is a bacterial universe again. And what does it take for this to become biologically available now, nitrogen? It takes a lot of reducing power, as you can imagine. Okay, you compare dinitrogen to ammonia, again, this is an enormous amount of electrons. And we need to do this because organisms cannot use dinitrogen in chemical reactions. Okay, it's very inert. So it has to be converted into this kind of form where it can be incorporated into the synthesis of all other things that we need, let's say amino acids, nucleotides, and so on and so forth eventually. It takes a lot of energy. You take something that's inert, something that's more chemically reactive, so you can imagine, and you take lots of reducing power, lots of ATP to do this. So what do you need? You need a special enzyme. You need a nitrogenase that can do it. You need a very strong reductant. Ferredoxin is one. And of course, you need ATP, right? As we saw here. So you have the enzyme, reductant here for the electrons, and the ATP. The last thing you need is the complete absence of oxygen. Okay? So this has to be done in a completely anoxic, anaerobic conditions. The enzyme is dead if you give it oxygen almost instantaneously in less than a minute. So how you get the reducing power? You can use a ton of NADPH if you want to, or if you are available, or if it's available to you, you can use photosynthesis again to make lots of reduced ferredoxin, and from then on you can go in these multiple steps, further reducing dinitrogen all the way to ammonia. By far the most biologically significant nitrogenase reaction happens in the roots of some plants, legumes especially, by a bacterium called Rhizobium. And it's a symbiosis of the bacterium and the plant. The bacterium lives in these root nodules, right, shown here and here, for example. And so the bacterium lives there. And as I said, it's not a parasite, it's a symbiosis, it's a true symbiosis. So there's a give and take. What does the bacterium take? It takes food, starch. It is fed by the plant, right? So the plant can make a ton of sugars, as we've seen with the Calvin cycle and the photosynthesis, and gives it to the bacterium, which lives in this nodule in the root. And what it does there is to give to itself, but a lot of it also to the plant, to give nitrogen, ammonia, nitrogen in the form of ammonia. It has the nitrogenase, and the reducing power comes in, and NADPH, you have ATP. The food is abundant. There is a lot of starch, as I said, from the plant. So the bacterium has the enzyme, and what it absolutely must happen then let's go back to the requirements. The bacterium provides a nitrogenase that the plant does not have. There is a lot of reductant. There is a ferredoxin here. And most importantly, there is a ton of ATP. You can do whatever you want, right? With unlimited supplies of food, starch that is provided by sugars that are provided by the plant. But the bacterium also has to take care of this, the anaerobic conditions. And this is what you see in this slide. It makes a kind of hemoglobin called leak hemoglobin from legumes. And just like our hemoglobin, it's very capable of binding oxygen. In this case, unlike our hemoglobin, which binds oxygen to transport it through various tissues that need it, here the leak hemoglobin binds it, but just sequesters it. So it's like a sponge of oxygen, this protein. It binds all the oxygen, takes it, keeps it really away from where the nitrogenase is. So the nitrogenase now can function because it provides an anoxic, completely anoxic environment for the nitrogenase to do its job and take dinitrogen and reduce it to ammonia. And as you can see here, this is mostly a bacterial universe. And our job here is simply to provide more ammonia into the system. That's all we do. Okay, we don't do too much.